All right, so yesterday we discussed tonicity and filled out that data table chart, right, about isotonic, hypertonic, and hypotonic. So now we're going to look at some examples so that you can figure out what's going on, because this is going to directly relate to the Salty Sailor lab that we'll be doing the next 2 days. So let's say this bag contains a 20% salt solution, all right? The water surrounding the bag is pure water, meaning that it's 100% water. So we could label the picture like this, yes? So in your drawings, label it like this. So the question is, what is the concentration of water inside the bag? If it's 20% salt, it must be how much water? 80% water. Excellent. That's pretty easy to figure out, yes? Awesome. Is the bag hypotonic or hypertonic to the water on the outside? Hyper, because hyper means what? More, right? Hyper means more, so it would be hypertonic. Tonic refers to the dissolved stuff in there. Is the water on the outside hypertonic or hypotonic to the bag? Very good, hypo. In which direction will the water move? Into the bag, right? From high concentration to low concentration. Where is the water in lower concentration? In the bag. So the water's going to want to go into the bag. In which direction does the salt want to move? Out of the bag, right? From high to low concentration. So the salt will move out of the bag. So what process might occur if too much water moves into the bag? It would burst. Do you guys remember what we called that in terms of cells from yesterday's notes? Or the other day's notes? Starts with a P. Close. Okay. Or excuse me, cytolysis. I'm sorry, I was thinking the opposite one. It's too early, right? Too much water goes into the bag, cytolysis. It could rupture. You guys were all correct on that part, right? It could get so big that it bursts. The movement of the salt and the water will continue until when? Excellent. It has reached equilibrium or they are equal on both sides. Good job, first period. After equilibrium has reached, what will happen to the movement of the molecules? They still move, right? But they still stay about the same concentrations on either side, right? So we could say movement will occur or will continue in both directions, but the equilibrium will be sustained. So we say water always moves from an area of High to low. Good job. Higher to lower concentration. In other words, water moves from the hypotonic side to the hypertonic side. I had a lot of questions about using these words in like an example, so hopefully this helps you guys realize how we use the language in order to describe the situations. All right, so I'm going to ask you guys to work on this with somebody next to you. I'm going to give you about 5 or 6 minutes to try and go through this, and then we'll correct it as a class, okay? All right, so go ahead and get started. Okay, so the bag contains a 40% sugar solution, so hopefully you put 40% sugar in there. The water solution surrounding the bag contains 40% sugar solution. So this is also 40% sugar, right? What is the concentration of water inside the bag? 60%. Does everyone know where we're getting that? 100% minus 40 is 60%. So this is 60% water, and the beaker has how much in it? What percentage?
60.
Also 60, right? It's also 60% water. So what types of solutions are these? Hypertonic, hypotonic, isotonic? Excellent. They're isotonic because we talked about iso means the same, right? Isosceles triangles have same sides. Isotonic means same tonicity. In which direction will the water move?
It'll stay equal.
Good. It'll stay equal. It'll go in and out, right? So the water moves in and out. Whoops. Sorry. And it's going to stay at the same rate, right? Some will go in, some will come out. As long as it's in equilibrium, it's a happy camper, and it'll stay right there. Does this help clarify this idea of tonicity to you guys a little bit? Hopefully. You can lie to me and just say, yes, I totally get it now. Wonderful. All right. I am going to pause the video again and give you guys like 2-3 minutes to work out the next problem together. All right? Go for it. All right. So let's look at this next example. First thing we got to do is label the drawing, right? So the bag contains a 5% salt solution. We know that the water surrounding the bag is a 25% salt solution, right? So what is the concentration of water inside the bag? Excellent, 95% water. What is the concentration of water outside? Perfect, 75%. Is the bag hypotonic or hypertonic to the water on the outside? Hypotonic. Why is it hypotonic? Excellent. It has less salt in its solution. So the water is going, on the outside is going to be what? Hyper or hypo? Hyper. Very good. Hypertonic. In which direction will the water move? Excellent. Outward, from high to low, right? Water will move out of the bag into the outside. In which direction will the salt move? Inward. Very good. Salt moves in from high to low concentration. Okay, what process might occur if too much water leaves the bag? This one starts with a P. It's in our notes. from a couple days ago. Plasmolysis. Okay, that's essentially when all the water goes out of a cell and it collapses on itself. Plasmolysis. The movement of salt and water will continue until equilibrium. Very good. Till both sides are equal. Excellent. After equilibrium has been reached, what will happen to the movement of the molecules? They keep going, but at the same rates, right? Very good. Good. I feel like you guys are understanding this. Do you feel like you understand it? Kind of? No. All right. That's a resounding no. So water always moves from an area of, good, high to low concentration. In other words, water moves from the hypotonic to the hypertonic. Very good. All right, facilitated diffusion. Now, I know that your Ed puzzle yesterday went over this a little bit, yes? Some of you were still confused about it, so let's run through this really quickly. Facilitated diffusion. Polar molecules, such as water or glucose, have difficulty going through the lipid bilayer of the membrane, okay? A polar molecule is anything that kind of has a charge to it. You'll learn all about those in chemistry next year. So for facilitated diffusion, there are transport proteins that help these molecules pass through the membrane more easily. For the most part, everyone got this right on the Ed puzzle, but I just want to reiterate. Facilitated diffusion means it's still going from high to low, but it requires a transport protein. Okay? High concentration, low concentration. There's the transport protein. Very exciting, I know. Polar molecules cross directly through the protein without coming into contact with the lipid bilayer. All right? It's like a little tunnel. That just makes it easier to cross through. This is known as facilitated diffusion because these proteins facilitate or help the diffusion of molecules across the membrane. Does diffusion require energy? No. Diffusion does not require energy because it's going from an area of high concentration to an area of low concentration. So facilitated diffusion, does that require energy? No. It's just another type of diffusion, okay? It is considered passive transport because the solute is moving down its concentration gradient. So if you see the word diffusion, facilitated or not, you know it's going from high to low. Facilitated diffusion really just speeds up the passage of a solute by providing a passage through the membrane. It does not alter the direction of the transport. It just makes it easier. Right? Like you could walk all around this school and there could be no hallways and you'd have to weave in and out of all the buildings and the rooms and the trees and everything, but hallways make it faster, right? Same thing with the facilitated diffusion. Okay? It just makes it easier to go across. active transport. Does this sound like it needs energy?
Yes.
Yes. Okay. So materials sometimes must move against the concentration gradient. So it's going from low to high. The cell often must move materials from an area of low concentration to an area of high concentration. Is that going with the concentration gradient? No. No, it is not. Does it, quote unquote, want to move that way naturally? No. All right? So we can say this is called active transport, and the cell must expend energy to accomplish it. If you have like a truckload of bricks and they dump it out at your construction site and you need to build a brick wall, okay, that takes energy. You have to take each one, put it into the wall. Same thing with active transport. You have to take it from a concentration, from a low concentration to a high concentration, okay? You're forcing it to go against the concentration gradient. This requires ATP. That is really important to realize, okay? What is ATP? What organelle synthesizes ATP? What organelle? Yes, thank you. Mitochondria, okay? So the mitochondria is making this ATP so that the cell can actively transport things that it wants into the cell and then release things that it doesn't want in the cell. So if small molecules and ions need to be moved across the membrane against the concentration gradient, it will require the use of protein pumps. Okay? Does a pump sound like it requires work to use? Yes. That's how you know it uses energy. Okay? These are embedded in the membrane, and the use of the protein pump requires energy. What form of energy? ATP. Good? So this is obviously a picture of the protein pump. All right. Now, large molecules may have been transported by the movement of the cell membrane itself. All right? So endocytosis is the process of taking material into the cell by a means of infoldings or pockets of the cell membrane. So it literally makes little arms, the cell makes little arms and hugs the material all the way until it just pulls it inside itself. Okay? The pocket that results breaks loose from the membrane and forms a vacuole within the cytoplasm. Large molecules and clumps of food are taken up this way. It just surrounds it and it's like, nope, you're mine now. And now it's inside the cell. Does that kind of make sense? So you can see here's the process. Cell membrane, it's reaching out, giving this brownie a hug. Okay, now the brownie's on the inside. Is delicious. Preferably with walnuts, just in case you're thinking of Christmas presents for me. Okay, moving on. And there's the vacuole. Okay, this requires a lot of energy. So the two types of endocytosis are phagocytosis and pinocytosis. I know, that's exactly what you were hoping you'd learn at school today. What are the two types of endocytosis? Oh, yes, love school. Woo. Phagocytosis is the engulfing of large food particles. When you bring me the walnut brownies at Christmas time. Pinocytosis is cellular drinking, okay? So when the cell engulfs a fluid. Okay? For example, if you bring me a latte with the brownie, I'll be all about this life, okay? Or a caramel macchiato, just throwing it out there. Exocytosis, what do you think this is? If endo means in, exo means out. This is cell barfing. No, I'm just kidding. Well, kind of. Exocytosis is the release of large materials from the cell. Kind of like cell barfing, right? You're like, oh, we don't need any of this. We're just going to put it out there. So a vacuole fuses with the cell membrane, forcing the contents out of the cell, as you can see in that little picture. All right, and that's everything.